So in this section, we're going to talk about the much more complex eukaryotic cell and the cell cycle and mitosis, which is how the cell is going to divide its chromosomes evenly between two daughter cells. So humans have 46 chromosomes. Dogs have 78. The humble honeybee has 32 chromosomes. So these organisms have got to assort these chromosomes so that each of the daughter cell gets an equal number and gets the right ones. So what is a chromosome? Each chromosome consists of tightly coiled DNA. And the DNA is coiled around histones, which are these proteins. When it's not tightly coiled, we call it chromatin. During the S phase, or synthesis phase, that we'll be talking about when we look at the cell cycle, the chromosomes are duplicated to produce sister chromatids. The sister chromatids are joined at the centromere. Before division begins. I want you to notice we've got some words that sound kind of alike. We've got chromosome up here. We've got chromatin, which is just the uncoiled chromosome, the same stuff, DNA and protein. And then we've got chromatid, and the chromatid, this is one chromatid, and this is the other sister chromatid. Okay, so make sure we keep those straight. The sister chromatids are identical. They're identical copies of each other. Okay, so let's look at the cell cycle. This is the series of events that takes place in a cell as it grows and divides. To grow and develop normally, an organism needs to be able to control how often it divides and the timing of its division. So our circle here represents the life cycle of a cell. And interphase, it spends most of its time in interphase. Goes all the way around here and then all the way to here. It only spends a short time in M phase. And I'm going to have to draw a little line, M phase. All right, so when it's in interphase, we divide interphase into three parts. G1 is the first gap. S stands for synthesis, and this is where the DNA is copied. So this is G1, and this is the S phase while the DNA is being copied, and this then is our second gap phase. And here the cell is preparing to divide. The second gap phase is followed by the M phase. The first part of the M phase is mitosis. This is where the nucleus divides. And then cytokinesis, where the plasma membrane, PM is plasma membrane, pinches in and makes two cells. So this is a drawing of a cell that's in interphase. Note that the DNA is in the chromatin stage. It's all unwound. We have a plasma membrane. There's a nuclear membrane that surrounds the chromatin. And we have centrosomes. These are the details of the meiotic phase. And the way I teach you to remember this is Penny Parker met a timid crocodile. And hopefully that'll help you remember. The first part is prophase, pro-metaphase, metaphase, anaphase, telophase, and cytokinesis. So Penny Parker met a timid crocodile. You can remember the stages of mitosis. So let's go and look at what happens in each of those phases. The first one we said was prophase. And in prophase, the meiotic spindle forms. Chromatin condenses to form the chromosomes. So when we had the cell that was in interphase, chromatin just looks like a big mess in here, right? When we get to prophase, we've got chromosomes consisting of 2 sister chromatids. As we get to the pro-metaphase, the nuclear envelope breaks down. The spindle attaches to the kinetochore within the centromere of the chromosomes. So each chromosome consists of 2 chromatids. There is a centromere right here in the center, and in the middle with the centromere there's a kinetochore, and that is what the spindle fibers, I'll make them green just so are going to attach to. And that's what you see happening here in this picture. The next phase is the metaphase. I always think of this one as they're meeting in the middle. So it's meta, and the chromosomes are now on a plate here. They're lined up between the 2 kinetochores. In the next stage, anaphase, the chromatids move away from each other. So we have this one moving toward there and this one moving toward here. Telophase, the nuclear envelope reforms, the chromosomes start to uncoil, and the spindle that's been pulling the chromosomes around disappears. The cytokinesis happens kind of at the same time, so I don't have a separate picture. But cytokinesis is where you get this cleavage furrow, and it's going to actually divide the 2 cells. Now in animals you get a cleavage furrow. In plants you get a cell plate. So plants will end up looking a lot squarer, and they'll get a plate that'll develop between the 2 cells, and then eventually gets to be. Okay, so that is mitosis.